Okay. You may have noticed while the lead-up time to the class was going on that I was running a slideshow of some of the images of DNA, and I'm not going to go through each one of these, but this gives you both a chance to see some of the structures that I've talked about in just additional representations that might make it easier to see. It also, I think, brings across how very beautiful DNA is, and it includes at least one or two examples of the wrong-handed DNA that I warned you off of. These pictures are not in your handout, but they are. This is one of the left-handed ones. It's wrong. It's labeled wrong. Don't make the mistake that the publisher did. But this is available, and I'm going to show you where it's available because I want to keep you up to date on some of the resources that are available for you at ICON. And so if we look, all right, no, that's not it. Goodness, let's see. Huh. All right. Well, ICON. Okay. If we go back to ICON, this is just there. You probably got an email. If you haven't, you will soon about Lynn's times and places. Once we have the room for Tim, we'll send that out as well. But the ICON website is a real resource for you for a couple of things. And so if you go to the content link, you'll see here articles that you might want to read about the origins of cells. We have an article on the hydrophobic effect for those who want more insight or a more physical understanding of that. And then these two articles, Genome 2.0 and Ode to the Code, are supplementary materials about DNA and the genetic code. Images of DNA is the slideshow that I was just showing right now. So if you want to look at those pictures in more detail, all you need to do is go to ICON, and those images are right there. Another resource that's a strong one on ICON are the links that we have. And in addition to being able to get to the Google movies for these things, I've been adding some additional links in here. When we get to the proteins, we'll look at the amino acids with JMOL. But there's also a mycoplasma genome applet, and I think that we may have a chance to look at a little later in the lecture today. So I'm just going to collapse that. Whoops, that's not what we wanted. I don't think. Nope, sorry. I wonder if I pasted the wrong. I did. That's my mistake. Ignore that for a little bit. I will get back to that and correct it. In the meantime, we do have the DNA. So let's just close that down for the moment. Okay, so let's go back to DNA. And I'm going to go to this to navigate quickly. Do people have any questions from our discussions on Wednesday that you want to ask about DNA? I think we got to the point of starting to talk about the genetic code. Is that right? Complementary and central dogma and that kind of thing. So let's just get down to there. Oh, this is so much better. I can't tell you how much better this is than last year's. It may sound like a small thing, but you have to live with a bad computer for a semester to realize. Do people have any other questions about the central dogma or the overall meaning for it, for information flow from DNA to RNA to protein? Okay. Let me get my pointer happy, and we'll be ready to roll. No, I didn't do that right. There we go. Okay. All right. Now, the fact that DNA is, when it's a gene, is present as a double-stranded molecule with this capacity for complementarity, the fact that hydrogen bonding provides a way to make a complementary molecule, is the basis for replication and the basis for transcription. In replication, it's fairly intuitive. You start off with, two parent strands, you make two new strands, now you have two complete copies. But in the case of transcription, it's a little more subtle. So let's take a little look at how complementarity works during transcription. You remember I said that DNA is an anti-parallel double helix. And one of the things that I like about this diagram is that it codes the two strands by color. So here's from 5' to 3' on one strand, And here's the 3' to 5' on the complementary strand. So the DNA itself is anti-parallel. The molecule in RNA polymerase is the molecule that's going to catalyze forming a complementary RNA molecule. That's what transcription is making a complementary RNA molecule. And the RNA polymerase is going to read only one strand at a time. It's going to read the template strand. Now, we're going to see that in some cases, either strand can be a template, but not for the same thing. So let's just focus on this as a template strand for the moment. The RNA polymerase is moving down, we can think of it that way right now, down that strand in one direction. And if you think of it in terms of the template strand, it is moving from the 3' end toward the 5' end of the template strand. But because the nucleic acids are always anti-parallel, the new growing molecule of RNA, the new messenger RNA, is being synthesized from its 5' end to its 3' end. So the 5' end is the oldest part of the molecule. The 3' end is the newest part of the molecule. So this kind of complementarity and anti-parallel synthesis is something that's going to underlie replication and transcription in both cases. We call the strand that is providing the site for things to be copied from the template, the one that's going to be used to generate the thing. And because the strand that's across from the template strand has the same sequence, except for U and T, as the new copy, we call it the coding strand. So there's the template strand that's used to make the new messenger RNA, and a coding strand that has the same sequence as the messenger RNA if you allow for T's and U's. Any questions about that? As you can imagine, if you are tightly hydrogen bonded like this, you can't slip in and make something new. This diagram is showing you that there's a little bit of unwinding at the site of transcription, and we can talk about promoters a little later in the course. So far, we've only talked about complementarity and the fact that A codes for T and G codes for C. And so you can make one DNA from another DNA, you can make a DNA from an RNA. Not a problem. So, you know, a DNA double helix can give rise to a messenger RNA single strand. Its 5' to 3' sequence will be the same, barring the T's and U's, as the coding strand of the original gene. But then we need to somehow or other make protein. And protein isn't made out of nucleic acids, and there's 20 amino acids. So there has to be some kind of a strategy for turning a linear sequence of 4 nucleotides, or 4 bases, possible bases, read off one after another, into a linear sequence of any of 20 amino acids. There's got to be some kind of a translation mechanism, some kind of a code that tells us how to turn nucleic acids into protein. And after brilliant experiments, and I don't know that we have time to go into them today, it was deduced that the nucleic acids can code for proteins if you take them 3 at a time. So the idea here is you take these 3 bases and make a complementary tRNA that is complementary to that. Those 3 nucleotides constitute a codon for leucine. So 3 bases equals 1 amino acid. CUC in the messenger RNA would be a GAG. In the transfer RNA equals a leucine. AGU in the messenger RNA equals UCA. In the tRNA equals a serine. GCC, CGG equals an alanine. So 3 bases, 1 amino acid. 3 bases is a codon, a single codon, and each codon accounts for 1 amino acid. This graph of the genetic code is one of the more interesting ones to my mind because it presents very dramatically the fact that the positions within a codon are not all equally important, and the fact that there are more than one way to specify a specific amino acid. So how do we read a graph like this? On the inner circle is the first nucleotide in the codon. Here's the second one, and here's the third position. So if we were to read this, we could see that UCC specifies serine. UCU specifies serine. UCG specifies serine. All four of these are equally good ways to say the amino acid serine. If I give you a sequence, you know reliably what that should turn into in amino acids. So we say that the code is unambiguous. Given a sequence of nucleotides, there's one and only one protein sequence that is the correct translation if you take them just as codons. We're not going to get into introns just yet. There's more than one way to say a given amino acid. That is, if you know there's a serine in a protein, you don't know what the nucleic acid sequence was. It could be any of 4 different or more things. So we say it's a redundant code. And there's a further characteristic of it, which is that it's resilient, and I think that'll become a little clearer on our next slide. It's not just that the triplet code allows us to take 4 bases and code for 20 amino acids. It's that the particular genetic code we have, the particular triplet code we have, is very resilient. It's very robust. Because if you make a change in the nucleotides, it may result in no change or a very small change in the protein. And let's take a look at that. Here is, and this, by the way, is in one of the supplementary handouts on ICON if you want the whole article from which this came. So look at this. There's 6 different ways to say leucine, okay? So mutating some of the nucleotides in there, you're still going to end up with leucine. But some of its neighbors are things like isoleucine or phenylalanine, so that not only are there several changes that still leave you with leucine, but many of the other changes would make you go to something else that's conservative. And similarly over here, that you can see that asparagine and lysine, for instance, are fairly close. And you have this code that has an ability to put in near synonyms. They're not exact synonyms, but near synonyms when you make a mistake. It's a very, very beautiful code. And the article that was exploring this did what's called a Monte Carlo simulation, where they just randomly drew out random codes and discovered that the code we happen to have only happens, a code as good as the one we have only happens 1 time in a million. That if you just randomly assign nucleotides to amino acids, you don't get this property of a beautifully resilient code. So this is an additional property that is very favorable to things going on about the business of living. Any questions about that before I move on? Okay, so that's... Now, part of what makes life interesting is that although all of our proteins are coded for in our DNA, there's a lot of our DNA that doesn't end up in a protein. In bacteria, most of the DNA ends up coding for proteins, but we're more complicated. And this is some of what it looks like when you look at higher organisms like ourselves. Let's say you start off in the DNA transcribing a particular gene. You have some gene that's about 250,000 base pairs long. This is not an impossibly large number. There are definitely genes this size and bigger. You transcribe it and you end up with a big piece of RNA. It has to be 1-to-1 because it's just been transcribed. So it's also 250,000 bases. Then in many cases, that transcript is spliced. That is, pieces of the RNA are taken out and what's left is stuck back together. I would compare it to splicing tape, but nobody has tape recorders anymore, so it's not going to mean anything. And so after the splicing event, there might only be a few thousand bases in the RNA. And of those few thousand bases, maybe only a third or a half of them actually have codons. This untranslated region here and this untranslated region there don't make protein. Now, they may have very important functions, and I can talk a bit about those later on, but they're not making protein directly. And so these, slightly more than 4,000 bases are giving us about 1,500 codons. Remember, you divide the number of bases by 3 to get your codons. And so these 1,480 codons can be translated, and now you have a protein of 1,480 amino acids. So you're starting off with 250,000 base pairs in the DNA and ending up with a little over 1,000 amino acids. This gives you a little bit of a feeling for our genome and just how dramatic that can be. Perhaps as much as 1/4 of the DNA is not even transcribed. And then of the remaining 1/4 that is transcribed, just this sliver, a few percent, or perhaps 1% in some organisms, ends up in a protein. There's another property about the central dogma that was not completely appreciated, or at least it wasn't dwelt on at the time that it was formulated, because they didn't know as much of the biochemistry as we know now, which is that the central dogma is what I call recursive. That is, you need what you're making to make what you're making. And let's just take a little look at that for a second. Information flows from DNA to make more DNA. Information flows from DNA to make RNA. Information flows from RNA to make protein. But to make more DNA out of DNA, you need RNA and you need protein. To make RNA out of DNA, you need RNA and you need protein. To make protein out of RNA, you need RNA and you need protein. And so there's this sort of embedded or self-referential character to the central dogma that for many years was sort of a fundamental mystery about how a system like that could ever have gotten started. I think later in the semester we'll have time to peek at a possible explanation, but it's an aspect of the central dogma that isn't usually dwelt on very much at the time. Other things that we can now do with DNA is to start to look at a chromosome in more detail, to look at a chromosome and understand its organization, where a specific gene is, how that location relates to other genes, and how rearrangements of genes can lead to diseases, okay? And this is giving you a sort of a taste of some of the ways in which genes and chromosomes can be mapped. Here is a schematic drawing of human chromosome 7. And it's showing you banded regions and non-banded regions. And the banding, this is sort of an example of a banding pattern. The exact basis for a band will depend on the staining method. It often distinguishes between active and inactive DNA. But there are a number of methods that are used. And this is showing you that the cystic fibrosis gene can be localized to a very small part of the human chromosome 7. So this is cytogenetic mapping. This is giving you a little bit of a taste of some of the kinds of rearrangements that are observed. These are using some of the older methods that just give you with a black-white, in effect, banding pattern. Using the newer methods I mentioned before, where you can paint a chromosome so the entire chromosome is a color, you can see here rearrangements so that this chromosome has a little piece that it's exchanged with this chromosome. So that's a translocation between those two chromosomes. The person holding these chromosomes has a complete set of information, but it's organized differently than it was in the parents and would probably be a problem if they try to have children. You can also have situations where you just have a translocation without complementary change. So a little bit of the blue one is over there now. If I remember correctly, one of these two is showing a comparison between human chromosomes and gorilla chromosomes. There's been a certain amount of shuffling around in the process of the evolution of those two species, but they're still close enough that the two sets of chromosomes react with the chromosome-specific dyes. We have developed techniques that make it possible for us to know exactly what the nucleotide sequence is of a piece of DNA. And this is pretty amazing when you think about it, because each of your cells has DNA this long in it if you stretched out. About a meter of DNA. You have about 3 billion base pairs of DNA. And yet we've developed methods that allow us to sequence gene along gene by gene. And how might that go? Well, let's kick in here at the point where we're looking at a single strand of DNA. If we want to sequence this DNA, we have to have some way of identifying this piece of DNA. And we use that a short piece of nucleic acid called a primer. The primer is going to be complementary to a known part of the gene we want to sequence. And in addition to that primer, we can add a DNA polymerase. So we're going to make more molecules of DNA. We're going to add deoxynucleotide triphosphates. So these are going to be the correct nucleotide substrates for a DNA polymerase. And fluorescently labeled dideoxynucleotide triphosphates. And we're going to see the structure of a dideoxynucleotide triphosphate in a second. So we have a primer that's bound to our target. And the DNA polymerase takes that free end of that free 3' end of the primer and adds on to it. And so it's making new DNA in that direction. Now, what is a dideoxynucleoside triphosphate? Well, if it's DNA, it had to be a deoxy at the 2' position here. But these are 2', 3' dideoxy. So this one is missing a 3' as well. So any place where this is inserted won't have a hydroxyl group at the 3' position for anything else to happen at. And so what happens is that the polymerase continues to sequence along until it inserts one of these dideoxynucleotides, because at that point it can't add a new nucleotide. And maybe that is a fluorescent A or a fluorescent G or whatever. By having the different dideoxynucleotides different colors, we can see that the chains that terminate with a C might be green, the chains that terminate with an A might be red, and so on. And at that point, all we have to do is read up this ladder of fragments to figure out the sequence of the DNA that we've just replicated. There are other things we can do with DNA. For instance, remember again, we've got this much DNA in our cells. We can cut at specific sequences within that DNA using an enzyme called a restriction enzyme. And restriction enzymes are called that because they're the enzymes that restricted certain bacteriophages from attacking certain bacteria. And so here's an example of extremely basic science that's turned out to be amazingly useful to all of us. So let's say we start off with a sequence like this. There might be some restriction enzymes that can cut here and here, and that gives you what are often called sticky ends, because obviously that bit can recombine with that bit with no difficulty. There are others that can give you what are called blunt ends, where it'll cut in the middle of the restriction site. Restriction sites are often 6 nucleotides long, sometimes 8, sometimes 4. Obviously, the length will determine how specific the restriction enzyme will be. The longer it is, the fewer examples of that there will be in the DNA. And then that gives us a chance to recombine things. So, for example, if we start off with a plasmid that's a good carrier and DNA from some source that we want to put into it, if we cut these pieces of DNA with the same restriction enzyme that has sticky ends, then when they're cut, they have ends that overlap, and some of the molecules will anneal to form something like this, where you have the original plasmid DNA and some new DNA that's now inside of it. And if we use a new enzyme called ligase, it can seal up those molecules, and now they're going to be intact, complete covalent circles. This gives you a little bit of an example of some of the kinds of plasmids that get used. It gives you a feeling for a couple of the properties that make plasmids more or less useful. This is called PUC18, and some of you may have already used it for experiments. P stands for plasmid. UC, because it comes from University of California at San Diego. So whenever you see a PUC plasmid, you can think Southern California and warm, mellow thoughts. 18 is just the 18th one they made. It has a number of features that make life more straightforward if you want to try to clone or change DNA. It has a multi-restriction site here. So this one has a site in the middle of this particular gene with 13 restriction sites. So 13 different restriction enzymes are potentially available to you to put things into it. It has a LAC-Z gene, which can turn things blue if that gene is intact. And it has antibiotic resistance. So you can select colonies that have had this put back in by whether or not they're resistant to antibiotics. So you don't have to look at every single bacterium in your culture. You can look at the ones specifically that have this plasmid in it. And on top of that, ones that are marked for having had an insert put into it. These principles of selectability and an easy site for insertion are common features among plasmids. Another technology among DNA that has really changed what we can do in genetics and in many fields within biochemistry is something called PCR. PCR is short for polymerase chain reaction. You can see the full name of it down there. And you're going to see why it's called polymerase chain reaction. In PCR, you start off with some kind of a target, and you have some kind of a primer that recognizes the thing you want to amplify. And so you melt it, you raise the temperature to melt it, you allow the primers to anneal, you then use a polymerase that can extend those primers, you melt it again, you add new primers, you extend that again. Well, let's look at what's happening then. You started off with one copy. At the end of the first round, you had 2 copies. At the end of the second round, you had 4 copies. At the end of the last round, you had 8 copies. So each round is doubling the number of copies of the gene. And so what we're seeing is the power of exponential growth. This is the same thing that underlies the pressure of populations or nuclear explosions. Whenever you have an exponential growth, you're going to have potential for very rapid expansion of things. I think it was Darwin who figured out that if every calf that every elephant had didn't die, the earth was going to be 30 feet deep in elephants in the course of some surprisingly short period of time. So exponential growth is a powerful tool. As a rule of thumb, 10 cycles will give you about 1,000-fold amplification. It's actually 1,024, whatever the last bits of binary are. So if 10 cycles is around 1,000, then 20 cycles is around 1,000,000. You can see where this is going. So PCR gives extraordinary power for amplifying DNA sequences. And what sort of things can you do then with PCR? You can use PCR as a way to generate a specific kind of mutant. That is, we have it in our power now to imagine in our heads a particular kind of mutant we want to look at, and then to make that mutant. So in doing that, you would have your original molecule that you're going to be making a mutation from. You separate the plasmid strands. You have primers in this case that have in it the mutation that you want, the thing you want to change. And you keep going through PCR, and after you do that more than a few times, almost all the copies are going to have the mutation rather than the original sequence. And you can select and identify that you in fact have the mutant you want. So if you can imagine now a particular kind of mutation you want, we have now the capacity to do that. A second technique that has benefited from PCR is pathogen detection. So for instance, there are some blood-borne parasites where you might have one or a few parasites per mil in the blood. This is not something where you can pick it up on the microscope by looking at it. Trust me on this. But PCR is enough more sensitive that it can pick up these very rare parasites that are nonetheless making a person very sick. The extreme sensitivity of PCR means that it's possible to identify DNA from a criminal. So on a crime scene, if you have somehow or other interacted with it, if you have left your fingerprints on it, if you have left spit or hair or skin, I don't know the fingerprints quite yet, we may not be quite that far, but certainly for the others I mentioned, PCR is sensitive enough that the DNA in that little bit of body parts that's left behind provides a sensitive marker for who's president. Now, we have this much DNA in us. It's obviously not going to be terribly useful to PCR the whole genome for a person. So in trying to identify individuals or in trying to do genetic studies, we didn't lose the signal. Good. Okay, I wasn't quite sure. It's better to focus on a restricted subset of that meter of DNA. And one of those restricted subsets that's turned out to be very powerful are something called RFLPs. And they get called RFLPs because their full name is restriction fragment length polymorphism. So let's break that down. A restriction fragment is simply a piece of DNA that forms when you cut with restriction enzymes. And so if you start off with your original DNA here at the top and you cut it with a restriction enzyme, a sample B that only has 2 sites in it will make one piece that's that long, and sample A that seems to have 3 sites in it will make 2 pieces that are shorter but add up to the same length. And so if you separate that DNA by gel electrophoresis, you can see that this one has 2 bands where that one had 1, and that the numbers add up, that this person's adds up to that. So PCR can amplify the DNA enough that you can detect RFLPs among individuals. And it can also be used to diagnose genetic conditions. If you know of a site in a gene that is a frequent mutation site for genetic disease, you can use that as a target to identify that. This is one example of an RFLP using actual human-type specimens. Here is the restriction fragments from the victim. And you can see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 15 major bands, maybe another 15 or so minor bands. Here's a blood-stained specimen that was found in the area. And you can see that this blood stain is not from this person. Yeah, it shares that band, but that's about it. Almost all the other bands are different here. And if you have 3 people who are suspects, you can see fairly quickly, looking at these 3 suspects, that only one of those 3 matches closely. Whether or not there's a good match between the particular blood stain that you have and an individual will to some extent depend on a population that you're looking at. That is, the frequency of a particular pattern in one group may be different than in another group. Italians may be different than Japanese. So you might want to know that you're drawing from equivalent populations. But if considerations like that are taken into account, the reliability of identification this way is just really quite high. It also raises questions about whether it's fair to simply do a mass screening. A few years ago, there was a series of deaths in a village in England, and the police finally decided to basically try to get DNA samples from every man in the village. And this was England, and they tend to be cooperative, and I think they ended up getting almost everybody to agree to give blood samples. But it does raise questions about is it appropriate to have, in effect, a search of your person without any real reason to think that this person might be guilty of things? Any questions so far? Okay. Okay. Well, all this ability to play with DNA, to use DNA as a tool, has made it possible for us to develop a number of organisms as being especially good targets or systems for asking questions in. And what they're good for is partly a function of the organism and partly a function of the question. I just want to introduce you to 4 of these model systems because they're ones that have contributed a great deal in different ways. One that I know you've all heard of is E. coli, and this is a scanning electron micrograph of those. E. coli is a fairly small cell, apparently fairly simple, metabolically fairly complex. It's a parasite. It likes to live off of us. It's not free living. But it certainly has been a very powerful tool for a lot of the fundamental kinds of metabolic questions and so forth, the basic biochemistry of how prokaryotes work. E. coli is a prokaryote. It doesn't have a nucleus, it has a circular chromosome, doesn't have a nuclear envelope, doesn't have any mitochondria. So there are a whole lot of questions that E. coli could never answer. So the next level of sort of complexity to start looking at you can see in a system like S. cerevisiae or Saccharomyces cerevisiae. These are the yeasts. They happen to be the yeasts that were used for beer making, and they're an example of why the Germans dominated biochemistry for the first half of the 20th century. Beer yeasts were one of the major sources for biochemicals, and the Germans had a lot of great breweries. I'm not making that one up. They had a really rich source of ATP and enzymes and so forth, and it was wonderful. So beer yeasts, or S. cerevisiae, have a number of nice properties. They are eukaryotes. They do have sex, sort of, so you can do genetics on them by crossbreeding them. They do have subcellular organelles. They're unicellular organisms. So what you're looking at is the biochemistry of a cell, of one eukaryotic cell at a time. You can't explore questions of development. And that's one of the reasons that another model organism called C. elegans came to be used. This is a light micrograph of C. elegans. C stands for Caenorhabditis. Don't worry about spelling it. And Caenorhabditis is a small worm which has the lovely property of being transparent. So you can look inside of it while it's developing and see what's doing what to whom. It also has the interesting property that it has very deterministic development, that you can track the history of most of the cells in the adult back to the original egg cells and know that these 4 cells at the 16-cell stage are going to go on and provide the thus and such. So it has provided a surprisingly powerful method for looking at development, even though they are really tiny worms. You can just barely see them with your eye. A fourth model organism that's worth being aware of is this one. It's green. As you can guess, it's a plant. It's Arabidopsis thaliana. It's a very, very, very small mustard plant. So an Arabidopsis plant is maybe about that big. It grows rapidly. It can go through a couple of generations a month or so. And it allows genetic detailed studies of all the things that are unique to plants, photosynthesis, development at the level of organs like flowers and leaves and so on. So all of these model organisms have given us very specific insight into particular areas of biochemistry. They remind us of the general strategic question that you need to pick your system to study for its strengths relative to your question. It may be much easier to grow S. cerevisiae than to grow C. elegans. But if what you want to know about is what genes control head formation, there's no point in growing S. cerevisiae. You have to work with a model that does do what you want it to do. I find this kind of interesting, looking at how many genes go into making up humans or other kinds of organisms. If we start from the smallest genome on this table, we have here Haemophilus influenzae, which is the bacterium that can cause a variety of human diseases. H. influenzae has about a little less than 2 megabytes of DNA, 2 million base pairs. Has about 1,700 genes. And this is a free-living organism. So this tells us that 1,700 genes is enough to make a living. You have a gene for about every 1,000 bases on one circular chromosome. E. coli is this partner that we've talked about just now. It's a commonly used model organism, about 3 times as much DNA, roughly twice as many genes. Pretty similar gene density, and again, a single circular DNA. Here's Cerevisiae. This is the yeast that we saw a picture of just before. It has less than 3 times as many base pairs of DNA than the E. coli. It's really not a lot more DNA. It has barely even twice the number of genes. So this shows us that a unicellular eukaryote is not that far beyond a prokaryote. It definitely needs more genes to be a eukaryote, but it's not dozens of times as many genes. It's about twice as many. On the other hand, it packs it into 16 linear chromosomes. So the organization of that DNA is quite different. And then when we start looking at some of these other organisms, we start to see some pretty rapid expansion. So here's about 100. These guys have about 100 megabases, 100 million base pairs, which is both that roundworm I just showed you and the Thaliana I just showed you. Fruit fly, about twice that. Nothing can actually fly. We get up into the mammals, and we're over an order of magnitude more still in terms of size. But that order of magnitude in terms of size doesn't necessarily translate into an order of magnitude more genes. This is kind of interesting. Look here, the plant has more genes than the fruit fly, like twice as many genes as the fruit fly. Less DNA, more genes. So in the case of Arabidopsis, the genes are packed fairly densely. And that shouldn't be too surprising. I mean, we don't have to photosynthesize. And we and rats have rather similar numbers of genes between us. We do somewhat different things with them, but not radically different in terms of the biology. We do radically different in other ways. And again, many more chromosomes than we had before. So, in the recent years, we have completed a map of the human genome, and scientists have gone on to map a number of other organisms as well to start comparing human genetic organization and human genetic composition to other organisms, and to better understand our own organization. Some of the things that made genome mapping a little bit tricky is that there is in the human genome repetitive DNA, where you'll see dozens or hundreds of copies of a sequence in a row in some places. Another is that some of that DNA turns out to be transcribed but not turned into genes. So how do you finally decide what the genes are in the human genome? There are several ways of doing that. We look for what are called open reading frames. So an open reading frame has the stuff at the beginning that looks like it should say start making a protein here, and something at a reasonable distance down that says stop. So it's an open reading frame. You could, in principle at least, make a protein out of it. Another way of identifying a gene is what's called an EST, an expressed sequence tag, that you take a pot of messenger RNA from a cell and make short tags that are complementary and see which of these pieces of DNA map to that. And the third way is by looking at their similarity to things that we know are genes. And by doing all of that, having the complete DNA sequence for humans, work is ongoing to completely annotate and interpret this genome. And this gives you a little bit of a sense of the complexity and density of genes in it. This is a figure from your book showing it. I'm going to see if the applet I opened works. Let me go down here and see. Let's see. No. DNA structure. Maybe it was in here. Let's see. Resources. Let's try this here. Okay. Oh, that's right. I made a mistake on that. I'm sorry. I did not map that correctly. I'll try to have that for the next map. I'll correct that link. Let me write a note to myself to correct the link. What it is it's an interactive applet, a little application run off of Java that has the entire genome of Mycoplasma genitalium, and you can toggle on and off different categories of genes and see how much effort it invests into transport, how much effort it invests into protein synthesis, and so on. So we'll just keep on going from now then. This is just showing you for E. coli. Now, we've been studying E. coli for more than a century. And look, 38% of them are still unknown. We don't know what the function is. This is like discovering that a third of Iowa is still unmapped. You know, what is going on? We don't know what that unknown 38% is. A lot of transport, bit of regulation, a lot of metabolism. Here's replication, transcription, translation. And I think we're at about 4:20, so I'll call that quits there. I'll be happy to answer any questions people might have.